So, you're getting ready to ace that advanced biology exam, right? What if I told you that memorizing complex cell structures absolutely doesn't have to be a grind? Today, we're stepping inside the ultimate microscopic factory. By the end of this explainer, you aren't just going to memorize a list of organelles, you're actually going to understand how they operate, how they integrate, and exactly how to dismantle the hardest questions your professors can possibly throw at you.
Here's the blueprint for our tour today. First, we'll establish the factory metaphor, trace the protein assembly line, check out power and waste management, flag the most common exam traps, and finally, we'll stress test your knowledge with some really expert-level questions. Let's get to work.
Section 1. The Ultimate Cell Factory. Establishing the Metaphor.
You know, biology gets infinitely easier when we map the microscopic to the macro world. So, consider this your ultimate cheat sheet. I want you to think of the eukaryotic cell as a highly efficient manufacturing plant. The nucleus? That's your manager. It holds all the blueprints but doesn't actually do any of the physical labor. The ribosomes are your floor workers, synthesizing the product. The endoplasmic reticulum is the literal assembly line, leading straight to the Golgi apparatus, which acts as your packaging and shipping department. The mitochondria serve as the power plant, and the cell membrane is your highly selective security gate.
Now, this factory design brilliantly illustrates why eukaryotic cells are just so advanced. By utilizing compartmentalization, basically walling off different chemical reactions into their own specialized membrane-bound organelles, efficiency just skyrockets. Contrast that with the simpler prokaryotic cell. Prokaryotes lack a true nucleus, and they completely lack membrane-bound organelles. They just rely on their circular DNA and ribosomes to get the job done.
All right, let's look a little closer at our factory security gate, the cell membrane. Described by Singer and Nicholson back in 1972 as the fluid mosaic model, we know this isn't just a static, rigid wall. It's a highly dynamic structure primarily made of phospholipids. Notice the role of cholesterol here. It's super important because it acts as a bidirectional buffer. At high temperatures, it restricts phospholipid movement, preventing the membrane from becoming too permeable and falling apart. Ultimately, this gate regulates exactly who gets in and out through active and passive transport.
Section 2. The Protein Assembly Line. Following the product.
Okay, so now that we have our static structures mapped out, let's actually watch this factory in motion and trace a single protein's journey. This is an incredibly high-yield integration concept for essays, by the way. Step 1. The manager copies the blueprint. DNA is transcribed to mRNA in the nucleus. Step 2. The ribosome workers read that blueprint and synthesize the protein. Step 3. It enters the rough ER for folding and quality control, helped by chaperone proteins. Step 4. It's sent over to the Golgi for post-translational modifications, like glycosylation. Step 5. It's packed securely into a vesicle. And finally, Step 6. It gets secreted out of the membrane security gate via exocytosis.
But wait a second. What about the smooth ER? While the rough ER is busy handling proteins, the smooth ER is more like a highly specialized wing of the factory. It lacks ribosomes, which is why we call it smooth, and it's entirely dedicated to lipid synthesis, glycogen metabolism, and crucially, detoxification via cytochrome P450 enzymes. Pro tip: if a question ever asks about drug detoxification in liver cells, the smooth ER is your immediate answer.
Section 3. Power and Waste Management. Keeping the factory running.
A factory this incredibly busy requires an immense amount of energy and a really robust cleanup crew, right? Otherwise, the entire assembly line just grinds to a halt. Enter the mitochondria, the power plant. This double membrane organelle generates roughly 30 to 32 ATP per glucose molecule through oxidative phosphorylation and the electron transport chain. But it definitely has some secrets. For one, it carries its own circular DNA, which is strictly inherited from the mother. Plus, if the factory is ever damaged beyond repair, the mitochondria will actually trigger self-destruction, apoptosis, by releasing cytochrome C.
And when it comes to waste management, examiners absolutely love to test your ability to distinguish between these two specific cleanup crews. Lysosomes act like the factory's recycling center. They use acidic conditions and enzymes like proteases to digest old organelles through autophagy. Peroxisomes, on the flip side, handle the chemical hazards. They perform beta-oxidation of fatty acids, and they neutralize dangerous hydrogen peroxide using a very specific enzyme called catalase.
Section 4. Common exam traps, where students lose points. All right, we've laid the groundwork. Now I want to show you exactly where students tend to lose points on advanced exams so you don't make the exact same mistakes. The crucial focus here is knowing your definitive distinctions. Do not mix up the rough and smooth ER. One is for proteins, the other is lipids and detox. Remember that prokaryotes might have ribosomes, but they absolutely do not have membrane-bound organelles, period. Lysosomes digest with acid, peroxisomes detoxify oxidative stress. And never, ever forget that the mitochondria is essentially a factory within a factory, complete with its own unique genetic code. If you master these traps, you practically guarantee yourself easy marks.
Section 5. Master the hard questions. Stress test your knowledge.
Okay, it's time to stress test your knowledge with some actual high-level multiple-choice questions. Let's see if you can apply those blueprints. Here's your first challenge. Why exactly does cholesterol stabilize the plasma membrane? Think back to our security gate metaphor and the dynamic nature of that fluid mosaic model.
Here's how this exact concept might appear on a real exam. Does cholesterol increase permeability, restrict movement at high temperatures, enhance protein transport, or bind directly to proteins? These distractors are specifically designed to sound plausible, and they're testing if you truly understand the biochemical buffering effect of cholesterol.
And the correct answer is B. Cholesterol acts as a bidirectional buffer. At high temperatures, when those phospholipids naturally want to spread out and become a bit too fluid, cholesterol steps in and physically restricts their movement, locking down the membrane's architecture and keeping our security gate perfectly intact.
Let's raise the stakes a bit with an integrated pathway question. Imagine a cell shows completely normal transcription, but it has defective secretion of proteins. Which organelle is most likely malfunctioning?
Looking at this integrated pathway question, you basically have to trace the entire assembly line in your head. Is it the nucleus, the Golgi apparatus, mitochondria, or the peroxisome? Well, if transcription is totally normal, the nucleus is fine. So we need to look at the step right before secretion.
The answer here is the Golgi apparatus. Think about it. If the manager at the nucleus is printing out the blueprints just fine, but the final product never actually ships out the doors, the breakdown has to be happening in the packaging and sorting department. The Golgi modifies and packages those proteins into vesicles for secretion. If it fails, the whole assembly line just backs up.
All right, one final expert-level conceptual question for you. If the inner mitochondrial membrane suddenly becomes permeable to protons, what is the absolute most immediate consequence?
When you look at the exact wording for this scenario, the options are definitely tricky. Does ATP production increase? Does the proton gradient collapse? Does NADH increase? Or does glycolysis stop? This is testing your deep, functional understanding of oxidative phosphorylation and chemiosmosis.
The correct answer is B. The proton gradient collapses. See, the entire power plant runs on the simple fact that protons are trapped in the intermembrane space, forced to flow through ATP synthase. If you suddenly make that membrane leaky, the protons just diffuse back across freely. The gradient is instantly destroyed, and oxidative phosphorylation immediately shuts down, halting ATP synthesis. The whole factory basically goes dark.
We'll wrap up on this thought. Compartmentalization increases efficiency. That is literally the true secret of the eukaryotic cell. Every single membrane, every organelle, every transport vesicle, it all works in perfect, isolated harmony to keep you alive. If biology has managed to build a factory this perfect, how might understanding its exact blueprints allow us to engineer microscopic biological machines of our own in the future? Keep studying, keep questioning, and I'll see you in the next explainer.